Welcome to my YouTube channel, Christy's Crash Course. Today we are going to continue on our endocrine series and we're going to attempt to answer much more questions, but focusing more on the thyroid gland as an endocrine organ. Stick around to see more. We're going to attempt today, it's a bit more of histological, but we'll also carefully walk our way from histological and we'll go so much so into the physiological aspect. So the question says, what is the embryological origin of the thyroid gland? And we have a outgrowth of the developing floor of the pharynx, outgrowth of the lateral pharyngeal wall, the outgrowth of the roof of the pharynx, and outgrowth of the developing heart. Which of the following doors are thyroid gland derived from? And so we're going to attempt to answer where other organs possibly also come from amongst the option. The option number D says outgrowth of the developing heart. If you look at this option, it is clear that it's usually the thymus that actually comes from that region. As such, it ends up in the anterior mediastinum. The option number C says the outgrowth of the roof of the pharynx. Remember, in our previous video of the pituitary gland, we saw that the anterior pituitary gland usually derives from an ectodermal region of the oral cavity, more precisely the roof of the pharynx. The outgrowth of the lateral pharyngeal wall, more so the pharyngeal wall, gives rise to tonsils. We could say more precisely the palatine tonsils. And our last option says the outgrowth of the developing floor of the pharynx. So this is our correct answer. The thyroid gland usually originates from the outgrowth of the developing wall of the pharynx, and then it grows downwards. It migrates by using the thyro. So there is a thyroglossal dot, which most of the time ends up being the isthmus of the thyroid gland. It says, what is the primary function of parafollicular cells in the thyroid gland? I like this question because it gives us a little insight into the Latin language. When we hear the word para, we must remember that it is quite Latin in origin and it is used to describe beside. And so when we hear the question, what is the primary function of beside follicular cells in the thyroid gland? So looking at this diagram also, we can see that we have the follicles here, or follicular cells, and then just around them, encapsulated in red here, we could literally see, so encapsulated here, follow my mark, we could literally see the presence of C cells which produce calcitonin. However, to look at other options, you could see that the thyroid gland, precisely follicular cells, will produce thyroxine, which is the active form of thyroid hormone. You could also see from this diagram, the hypothalamus releases the thyrotropin releasing hormone, while the anterior pituitary gland, specifically, if we want to be specifically the thyrotropes, produces what we know as the thyroid stimulating hormone, which goes to the thyroid gland and causes for the synthesis of T3, T4, which are basically thyroid hormones, mostly the active forms of the thyroid hormones. Our third question asks us, what is the main characteristics of the thyroid tissue? Remember that tissues are groups of cells, so when we hear the word tissue, we are mostly just saying groups of cells. What is the main characteristics of the structure of thyroid tissue? Represented below, we have different organs represented. This represents the lungs, this represents the pancreas, and this represents our thyroid. The functional and structural unit of the lungs is usually the alveoli. The functional structural unit of the thyroid gland is usually known as the follicles. Now, if you could follow my hands, you could see that these bulb-like structures, so each of these are cells, and then they encapsulate a fluidy region right inside, which we love to call the colloids. And then just outside we are seeing some cells here that are being embedded beside the follicular cells. So the answer to this question will be the follicles. Question asks us a similar question. It says, what type of cells are found in the thyroid gland that produces iodothyronine? So by now we've established that within the thyroid glands we find basically two types of cells. The first cells we find is the C cells. We also find the follicular cells. And we know by now that the follicular cells usually want to produce our thyroid hormone, and our C cells usually wants to produce our calcitonin. And calcitonin is important because it helps to decrease blood calcium level or serum calcium levels. Okay, however, something else I really want to quickly touch out for us is that we usually identify the thyroid hormones as T3 and T4. But much so, another name we can give to T3 is called 3,5,3' triiodo. So triiodothyronine. And why is this name important? Because it denotes the structure for us. So quickly, if we're looking at it quite biochemistry, in the view of biochemistry, we will see that our hormones are not all delicate as we want it to be. It's actually quite a simple structure, and when we see it from that limelight of how simple a structure it is, we gain better understanding as to this function. So if we say this is the benzene ring, so we'll call this A and B benzene ring. Now, what do we mean by 3,5? So this is 3. At this 3 point we have a iodine molecule. At this 5 point we have a iodine molecule. So this again will be 3' because we can't name the both of them 3. So 3, 5. This is 3', so this will be an iodine molecule. So if we have something like this, iodine here within the inner benzene ring, and then one iodine just outside within the 3 at the third position of this benzene ring, we call it the 3,5,3' triiodothyronine. However, for T4 is quite a different case. It has a lovely name, and it's called 3,5,3', 5' iodothyronine. What do we mean by this? It means that we just simply add an iodine to the 5th prime region. So this is 3', 5', 3 and 5. So in this case, when we add this iodine to the 5' region, this is now our T4. Very important. So to answer a question, what types of cells are found in the thyroid glands that produce iodothyronine? That will basically be follicular cells, because they are the only cell that will help in producing thyroid hormones. And remember, we said iodothyronine, these are just groups of thyroid hormones that have iodine bound to them, and of course they have the tyrosine residues. The next series of questions that we are going to answer, we need a basic understanding of the synthesis of thyroid hormones. Because of that, we are going to trace down what happens when our body needs thyroid hormone. What are the processes that occurs? In other words, how is thyroid hormone synthesized? Okay, so there is something called the hypothalamus pituitary thyroid axis, and it's just a circuit that enables, so you need to write that down. The hypothalamus pituitary thyroid axis is a circuit that ensures thyroid hormone homeostasis. Okay, so it's a circuit that ensures thyroid hormone homeostasis, and it's literally represented in this diagram. You could see that we have different neurons, but then we are going to focus solely on thyroid hormones here. And so more specifically, if you take an in-depth look into our diagram here, if you take an in-depth look into our diagram, you could see that the thyroid releasing hormone, so thyroid releasing hormone is put into the hypothalamus hypophyseal portal system, and the thyroid releasing hormone, which comes from the hypothalamus, precisely from the nucleus in the paraventricular region of the hypothalamus, releases thyrotropin releasing hormone. Thyrotropin releasing hormone goes through the hypophyseal system and it gets to your anterior pituitary gland. Now within the anterior pituitary gland, we have our cell which we call the thyrotropes. The thyrotropes receive this thyrotropin releasing hormone and causes for the release of thyroid stimulating hormone. So again, we have paraventricular region or nucleus, we call them. So this is the first step. The second step is that we have TRH is released. This is the second step. The third step, we have that after TRH is released, it goes to the thyrotropes and there's a release of TSH. So this is the third step. Very important steps for us to remember. Now, TSH or thyroid stimulating hormone has been released. What goes next? The TSH goes to the thyroid gland and it goes to stimulate. So it goes to anchor on its receptor. More specifically, if we want to be a little bit detailed into how the TRH helps in the activation of the thyrotropes, you could see from this diagram here that TRH goes to the second messenger receptor and more specifically the GQ coupled receptor, and you could see it goes to activate your phospholipase C. Phospholipase C helps in the conversion of PIP2 into DAG and inositol phosphate 3. And eventually DAG goes to activate your protein kinase C, but more so the very important player in this process is the IP3, because it goes to anchor on our inositol 3 receptor, usually present in the smooth endoplasmic reticulum. Okay, and then it goes to release calcium. Calcium is an important player in the exocytosis of molecules from the cell into the extracellular space. And so you could see this is our thyrotropes helping in the release of TSH. However, when TSH comes to this point, you could see that it goes to a receptor molecule, right? And the receptor molecule is called the TSH receptor. TSH binds to this receptor molecule, and now the beautiful dynamics of this TSH receptor is that it has a dual functionality. What do we mean by that? It has both a G alpha stimulatory protein, and it also has a GQ stimulatory protein. And we will go to see a beta diagram where it is being typified, where TSH causes the activation of both G alpha stimulatory protein, which goes on to activate AC. AC is usually called adenine cyclase. Is a very, very important enzyme in the study of hormones. And so adenine cyclase is usually activated to help in the conversion of ATP to CAMP, which goes further to activate our protein kinase A. And so we're going to look at that in the next diagram, how this process come to function together. Looking at how TSH has a dual functionality, this diagram gives us a better understanding of what is going on. And so you could see TSH has gone beyond the pituitary gland, and it has come to our follicular cells. Now this is the cell membrane of this follicular cell, so the cell membrane is just here, and it binds to TSH receptor. So this is thyrotropin stimulating hormone receptor. TSH binds to it, and you could see that we have the G stimulatory and the GQ protein. The GQ protein will go further to activate our phospholipase C. Phospholipase C. Phospholipase C, when activated, helps in the conversion of PIP2 to give us DAG and inositol phosphate 3, right? But the entire dynamics of these activations is such that they help in realizing each and every one of these functions. So they increase the iodide apical efflux, they increase the hydrogen peroxide generation, they increase the thyroglobulin iodination, they increase the activation of constitutive nitric oxide synthase. More so, if we look at it from the aspect of the G stimulatory protein, you could see that it goes further to activate adenosine cyclase, a very, very instrumental enzyme. And adenine cyclase converts ATP to give us CAMP. CAMP goes to convert our inactive protein kinase A, and we have an active protein kinase A. You could see literally the phosphorylation process that took place in ensuring the inactivation becomes an activation. And more so, we see that this protein kinase A goes to increase sodium iodide synthetase expression, thyroglobulin expression, and thyroid peroxide. So the next question to be answered is, what are these synthetase? What is thyroglobulin? What do you mean by what really is happening in this whole process, right? And so we'll go further to explain what these channels, what these synthetase or co-transporters are, and then what is thyroglobulin, because it is a reoccurring word we will get to hear in the study of thyroid hormone synthesis. You could see that this is tyrosine. And remember that tyrosine is an amino acid, right? So this is the tyrosine structure. And so when we talk about thyroglobulin, this is the structure. So it's a continuous long chain we call it a glycoprotein, and it's usually synthesized by the endoplasmic reticulum, precisely the rough endoplasmic reticulum, furthermore packaged and modified in the Golgi apparatus, and eventually sent out from the follicular cells into the colloid of the thyroid gland. So just to give us an imagination of what is happening. And if you look more so at these 3 and 5, in our previous slides we saw how T3 represents it represents triiodo 3 5 then 3 prime, right? And so you could easily have a picture of what will happen in the process of iodination, meaning that in the process of adding iodine. So if we were to add iodine to this point and this point, you will quickly understand that, so it means we will get maybe a diiodothyronine. If we just add iodine to this point, quickly understand that, so we could get a monoiodothyronine from this structure. And then when 2 of them come together, a di and a mono will eventually form T3. And if 2 diiodothyronine comes together, will eventually form. So if we could imagine that this was, this was also a diiodothyronine, right? So if these two decided to come together, we will form a thyroxine. All right, so just to give us an imagination of what happens in the process. So now we are going to have an overview of everything happening all at once at the follicular cells. We're going to look at the channels, we're going to look at the transporters, and eventually we're going to see how it goes from TSH eventually activating the transporters and also enabling the thyroglobulin formation, to how we eventually get our T3 T4 at the long run.